Welcome to Biology class. Today we are going to discuss the chapter namely Biotechnology, Principles and Processes. This is the first chapter of Biotechnology unit of 12th class. And in this chapter we are going to discuss what is meant by biotechnology, what are the principles of biotechnology, what are the tools essential for recombinant DNA technology, and then the process of rDNA or recombinant DNA technology. As you know that one, there are a lot of branches of biology is there to support the humankind or the mankind or human society by providing the products and services, etc. Biotechnology is also such a kind of branch which is providing and still going to provide many type of products for the human society or the human welfare. And here, if you are taking the biotechnology, we can see the modern and as well as the ancient biotechnologies. In the ancient biotechnology, what we are doing means we are usually using the entire group of organism, like we are using the yeast or the microbes for the preparation of bread, wine, and as well as curd, etc. And at the same time, we are using the exact product of the gene or the part of the gene for the betterment of the human beings also. For example, in vitro fertilization and the gene therapy, etc. are example for the modern biotechnology. And here we can see one definition, simple definition regarding what is biotechnology. It deals with the techniques of using live organism or enzyme from the organism to produce products and the process useful to humans. So we are using either the organism or the product from the organism as in the form of the product or the services. At the same time, according to European Federation of Biotechnology, there will be a clear-cut definition about the biotechnology, and it states that it is the integration of natural science and the organism, the cells and parts thereof and molecular analogues for products and services. And here also we can see the integration of natural science and as well as any organism or the cells of that organism or the parts or the molecules of that organism for the products and as well as the service of the human beings. This is about the definition of European Federation of Biotechnology. Coming to the point of the principles of biotechnology, we can see there are two core techniques are there, two major branches are there that enable the birth of biotechnology. One is called as genetic engineering, another one is chemical engineering. In genetic engineering, all the techniques we are using for altering the genetic material of an organism, either RNA or the DNA, and to insert that particular altered gene into the organism. At the same time, after that, we want to get the product from the altered gene or the organism itself, for which we are providing a sterile ambience in the chemical engineering for the growth as well as getting the product of the organism which is having the altered gene. So genetic and chemical engineering are the two major branches or the techniques that supports the birth of biotechnology. And if you are taking the history of biotechnology, especially the rDNA technology, and we can remember the two great scientists, namely Stanley Cohen and Herbert Boyer. In 1972, they constructed the first recombinant DNA. They have used two organisms, one is the Salmonella typhimurium, another one is E. coli bacteria. What they have done means, they have taken the antibiotic resistant gene from the plasmid of Salmonella typhimurium, they cut it and removed, and after that one they have ligated that part of antibiotic resistant gene with the plasmid of other bacteria. So here for cutting and ligating they have used different type of enzymes also. After that, the prepared plasmid which contain the rDNA or the gene of interest, they inserted into another bacteria, namely E. coli. So here the three activities we can see here. One is cutting and taking the gene of interest and ligating that gene of interest into the plasmid, and then it is inserting into organism. This one they have done, and based on which we can see that one. Here the three basic steps are there for the recombinant DNA. One is identification of a desirable gene. Stanley Cohen and Herbert Boyer, they identified an antibiotic resistant gene from the Salmonella typhimurium. And the second one is introduction into the host. E. coli was the host in which they inserted the DNA gene of interest with the plasmid. So the plasmid is a vector which carry the gene of interest. And the third one, maintenance of a desirable gene and its transfer to progeny. That means the chemical ambience what we told earlier, and in which they have inserted the organism and the organism is growing and getting the product. So these are the three major steps in the creation of rDNA or recombinant DNA. One is identification of desirable gene, introduction into the host, and third one is maintenance of desirable gene and its transfer to progeny. We can see here the tools of recombinant DNA technology. What are the key tools essential for accomplishing or the creation of one recombinant DNA? We can see here restriction enzymes are there, polymerase enzymes are there, ligases are there, vectors and the host organs. Clearly speaking, we can see the restriction enzyme, an important tool for the creation of rDNA. Another one is cloning vector and a competent host. And the first one we are using for cutting and taking for the gene of interest. Second one is carrying the gene of interest. Third one receiving the recombinant, etc. And this is the picture representation of the entire program of the rDNA. Here we can see again the mechanism of the creation of one organism, and as well as here we can see the gene of interest we are taking from here and the plasmid we are taking, and here the ligation by cutting by using which one of the restriction nucleases, and after that ligating here, and this is the recombinant DNA inserting into the host, etc. This is the host and this is the vector, this is which one gene of interest, and different type of enzymes we are using. In detail we are going to discuss. Restriction enzymes. We know that these are the peculiar type of enzyme which can be seen in the case of prokaryotic group of organism, and it is a defensive mechanism in the case of prokaryotic group of organism to prevent the entry and the multiplication of especially the viral or the viral particles or the virus, etc. And at the same time, the same we are using for the recombinant DNA technology also. Here we can see chemically these restriction nucleases are the chemically they are nucleases, the enzyme which can cut or the cleave genetic material, either RNA or the DNA. And here the nucleases are of two types we are telling based on their activity. So they are called as endonucleases and exonucleases. The function of the nuclease is to cut the DNA or the genetic material. Where at which location they are cutting, based on which, what type of genetic material they are cutting, based on which we can classify them as endonuclease and exonuclease. As it indicates, endonucleases are the restriction nucleases which will cut within the DNA. At the same time, the exonucleases cutting at the ends of that particular DNA also. And how they will cut, how they will select, we can see again, and here we can see recognition sequences. And these enzymes will read the DNA and they will identify a particular nucleotide sequences, and that particular area they will read and they will cut. That kind of area, every resistant enzyme will identify different sequences is called as normally the recognition sequences. That is the area they will cut. And the area will be always in palindromic patterns also. Palindromic means the word which you can read in both the directions. For example, we have given here the example for the palindromic sequence of ECO or first or ECO or 1, then bacterial in restriction enzymes also. Here you can read here the letters as GAATTC in one direction, and here the next direction we can see again GATTC, the direction will be there. In that particular area only what is happening, they will cut also. And here the restriction enzyme, there is a nomenclature of enzymes are there, naming of enzymes are there. How it can name, for example, we are taking ECO or first comes from the organism Escherichia coli, and what does it indicates? We can see E indicates, the first letter indicates the name of genus. The remaining two letters indicate the species, that means Escherichia coli. And R indicate this is the organism from which the strain of that bacteria from which we have extracted the enzyme. and the Roman letter, the first or the second, 1 or 2, etc., is an indication of the order in which we have extracted that particular enzyme from the organism. And here the chart shows that one type of restriction enzymes extracted from the different group of organisms, and here we are showing the target sequence or recognition sequence of the concerned enzymes also we can see. So naming is like this, and the first letter indicate the genus, second one indicating the species, and next the capital letter indicate the strain, and the number indicate the order of extraction of enzyme in that particular strain, etc. Next, the mode of activity of that particular enzyme, etc. Normal level mean. What they will do means they will cut the restriction endonucleases cut at a particular patterns of the nucleotide bases, what we call recognition sequences. And here we can see they will cut just in the middle of that particular part, leaving them as a hanging end or the sticky ends will be there. Here we can see in between G and A, both the direction they will cut. So that separation of the strand will be taking place by keeping a sticky ends. Here we can see the sticky ends. The same will be utilized for making or fixing with the opposite or the gene of interest if it happens. Like we can see, to cut both the gene of interest or the foreign gene and as well as the plasmid, we are using the same restriction enzyme. Otherwise, we cannot fix them with each other by using the sticky ends, etc. Will be that this is the activity of the enzymes, etc. Next one, after this one, cutting and making or separating one. There is some techniques are there to get the separation of the DNA strands, etc. What we are using the technology, as you know, that one it is the gel electrophoresis. The fragments after cutting with the restrictions are separated by using the gel electrophoresis. What we are doing in the controlled condition, in all the condition requirements, we are keeping in this is the electrophoresis machine itself, and which is having an electric field also, in which these are the port in which we are adding the liquid which contain the suspension which contain the DNA fragments, and when it is applying the electric current itself, due to the negatively charged condition, it will move towards this area, it will move towards the anode also, so that based on which we can identify the fragments of DNA. And to get the exact result, to see the things, etc., we are doing some more things. That means addition or treating with a particular stain, namely ethidium bromide, and after that one, we are exposing the ultraviolet radiation so that we can get an image of the DNA fragments here. And from this gel or the matrix, etc., we want to separate them for the further activities. And that kind of the separation and as well as taking out from the gel matrix is called as elution. So elution is the separation and extraction of DNA bands from the agarose gel. It is a matrix or the medium of electrophoresis. And this mechanism is there to identify or separate one normal level separate one the DNA fragments from the medium, etc. So we have discussed the restriction enzymes and their activity. Right now we have separated them also. And we can see in the case of the second one, we can see the cloning vectors are there. Cloning vectors means the vectors are normally carrying the other organisms. Generally, biology we are discussing the vectors. Mosquitoes are the vectors which will carry the pathogen. Like to carry the gene of interest, we are using some of the other genetic material to carry them. They are called as normally cloning vectors. Generally, based on the number of the production of the copies, we are taking the plasmid. Plasmids are the extra chromosomal rings we can be seen in the case of prokaryotic or the bacteria. And at the same time, bacteriophages, viral genome also we are taking for this purpose by inactivating the other part in the case of bacteriophages. And both of them are having the ability to replicate within the cell, host cell, in more number. So let us discuss what is the features of a cloning vector should have to perform the activity. Here we can see the three major features are there should have a genetic material to carry or to function as the cloning vector. One thing is that one important part is the origin of replication. As the diagram indicates, it is the plasmid of PBR322. And here we can see the ORI or ORI or origin of replication, from where replication starts. Another one is the selectable marker. Remaining, these are all the genes only of that particular DNA, and these are all indicating the type of the enzyme we are using to cut that particular area because of their recognition sequence, etc. So the first one is origin of replication. Second one, selectable marker. These genes help to select recombinants over non-recombinants. After the mechanism of the formation of normally the rDNA, we want to identify them as the recombinant and non-recombinant. Thereby, we want to separate them transformants and non-transformants. By using the activity of the particular genes, we are using all these things are, for example, ampicillin and tetracycline resistant genes can be used as which one are the selectable marker. Same time, the cloning site is another one. Cloning site, these sites refer to the recognition sites of restriction enzyme. And here we are using the same enzyme, we are cutting and we can insert the gene of interest to that one also. So based on which we can use them as the selectable marker and as well as the cloning site. So remember, the three important features of a vector should have is origin of replication, another one is selectable marker, next one is the cloning site. Next one important thing is the competent host, the organism which can carry the recombinant DNA. Normally, if we want to insert the same thing into the organism, for example, the bacterial cell, the fungal cell, plant cell or animal cell, anything can become the host itself. At the same time, the problem is that the recombinant DNA, as the name indicates, it is DNA or deoxyribonucleic acid. It is hydrophilic in nature. We want to insert that recombinant DNA into the inside the cytoplasm of the host cell. But the problem here is generally the cell wall will not take up the recombinant DNA because of their hydrophobic in nature. So hydrophilic components cannot be absorbed by, will not be absorbed by the hydrophobic cell membrane, for which we want to make them to competent. It is called as a competent host for the transformation. So we will make them to absorb the gene of interest from the external medium. So there are 4 different activities are there to make them to competent, and we can see the chemical treatment and incubation, and next one is the micro injection, next one is biolistic or gene gun and disarmed pathogen. All these techniques are using in different organisms also. In the case of the chemical treatment and incubation, we will take the gene of rDNA in the medium in which we are introducing the bacteria or the organism which are not having the plasmid, and we are making them to take up. How it is possible? Different methods are. We are treating the liquid or the solution with the divalent cation like calcium. After that, we are keeping them in the very cold condition. Then we are keeping them in the high temperature. Again, we are changing it to which one the very cold temperature or the ice, etc. Like a heat shock method we are approaching here. By the time the cell membrane will be ready to absorb the genetic material from the external one. That is taking place in the case of chemical treatment and incubation. Second one, micro injection. As the name indicates, it is the direct injection of the rDNA into the cell. It can normally see in the case of, we are employing this method in the case of animal cell to introduce the rDNA. At the same time, the biolistic or the gene gun, normally we are using in the case of the plant cell. Directly we are inserting with the high velocity into the plant cell, in which we are using the tungsten or the gold microparticles, and it will be covered with the rDNA, etc. we are doing. And in the case of disarmed pathogen, again we are inactivating some of the genetic material part of the organism, and then we will insert also. So like the different techniques we are using for inserting or introducing that particular gene into the host organism are there. For example, chemical treatment and incubation, micro injection, biolistic or the gene gun, and disarmed pathogen. And these are the 2 images of micro injection and as well as biolistic or the gene gun, in which directly we are inserting here, and here we are bombarding them with the microparticles bombarded with the target plant cell, etc. are there. And these 2 things are using here in the animal cell. This will be in the case of which one the plant cell we are using also. And this is the entire process. Normally we are taking place at the time of the creation of a genetically modified organism. Here we can see the foreign DNA is there, and here the vector is there, and we are cutting by using the nucleases enzyme, and both the structures foreign DNA as well as the vector DNA should be cut with the same restriction enzyme to make them sticky ends. After that, we are ligating by using the DNA ligase, and here we can see the recombinant DNA, and we are inserting into, this is the host, these are the methods, so that it will insert and it will absorb, and after that it will provide and then all the genetic material will be transferred again. So this is the general activity which are taking place. Let us see what are the important processes which are taking place to creation of the material. So here we can see the process of recombinant DNA technology. There are different steps. The first step is isolation of the genetic material. Second one is cutting of the genetic material, and third one is amplification of genetic material by the polymerase chain reaction. Next one is insertion or introduction of the genetic material into the host body. Next one is getting the product of the rDNA or the gene of interest, and the last one is separation or the purification, namely the downstream process, etc. Let us see what is happening here, what are the requirements essential. Here we can see the isolation of genetic material. If we want to identify, if we have identified the gene of interest and the plasmid also, it may be of different types of the cells. And if the identified gene or the plasmid, if it is there in the plant cell or in the bacterial cell or the fungus cell, we want to isolate that gene of interest by digesting their cell wall, for which we are using lysozyme for the digestion of the bacterial cell wall, chitinase for the digestion of fungal cell wall, cellulase for the digestion of the plant cell wall. After that, we are taking out the genetic material, and which will be treated with the new proteases and RNAs to remove the protein and RNA along with them. And after that, treating them with the chilled ethanol to avoid all the other impurities, etc. This is the first process of isolation. After that, cutting of DNA at a specific location. As we told earlier, the cutting is done with the help of restriction enzymes. After that, we will separate them by using the gel electrophoresis. After that, we will ligate them. The plasmid with the gene of interest will be ligated by using the DNA ligases. All these activities are taking place during these steps. Cutting, separation and formation, etc. will be taking place. And at the same time, the next level is amplification of the gene of interest. If we want to get a more number of copies of that recombinant DNA, we can use another technique, namely an artificial technique, namely polymerase chain reaction. And it has developed by scientist, namely Carey Mullis in 1983. And an artificial medium, by providing all the requirements, we can artificially produce more number of the same copies. How it is possible? We can see the three major steps, namely denaturation, annealing and extension. In the next slide, we can see the process here also. Here, the first process is the denaturation. And this is our recombinant DNA. And this recombinant DNA, if one copy is there, we want to keep them in the particular temperature. And you know that one, they cannot tolerate the high temperature. But at the same time, if we are keeping them high temperature in between 90 to 70 degrees Celsius, so that what is happening, there will be the denaturation, the separation of the double stranded into single stranded ones. That is happening by the process, namely denaturation. And after that one, we can see the two strands will be formed as each template strand, single stranded DNA. And this will be the template for the formation of the next strand, for which we are using the primers, and as well as which one of the other conditions like DNA polymerase and deoxynucleotides, etc. But the annealing is nothing but the joining. What we are doing, these primers will join with the template strand, and they will start the extension or elongation of the new strand by using the DNA polymerase and deoxynucleotides. Here we want to remember one important thing, that this is an artificial means which are taking place at a high temperature. Generally, DNA polymerase cannot tolerate high temperature. So that what we are using right now means we are using a special thermostable DNA polymerase, namely TAC polymerase, which have extracted from the Thermus aquaticus, one bacteria which are living in the hot springs or the streams, etc. High temperature of the water. So they can sustain, they can tolerate the activity, and they were doing the normal activity of polymerization also. So this is the extension also. After that, what we are getting, we will get a lot of same type of DNA fragments will be available in the case of this group of mechanism also. So the method is called as polymerase chain reaction. So we are getting a lot of billions of the copies will be available within short time also. After this one, the next process, as we told earlier, discussed earlier also, the insertion of the recombinant DNA into competent cells. Already we identified the cell, whether it is the plant or the animal or the bacteria or the fungi. By using the different methods like chemical treatment and incubation, like micro injection, like biolistic or the gene gun, or the disarmed pathogens, etc. We can insert that particular gene of interest and along with the rDNA into the organism also. And we are providing other conditions essential for them. Then the next stage is obtaining the gene products. This is a very important one. We have done all these activities to get the product, like any therapeutical product, or in the case of enzymes or the alcohols, whatever the product are there, we want to take them, for which these genetically modified organism or the host organism which have prepared will be kept in the particular condition or in the culture medium. So this kind of the product what we are getting from genetically modified organism, we are calling them as recombinant proteins. The product which are getting from the heterologous organism can be called as the recombinant proteins also. And these proteins, how we will get it, obtaining the product. We are culturing these microbes in the controlled condition in the large vessels, etc. These vessels where we are culturing the genetically modified organism to get the product are called as bioreactors. These bioreactors from 100 to 1000 liters of capacity, and which is having very controlled conditions, which provide all the requirement for the growth and as well as multiplication of which one genetically modified organism also. And here the two images, types of two bioreactor image they have given. Steamed type and sparged type of bioreactors are there, which are commonly using in the commercial level for the production of the product in the commercial level also. And this bioreactor will provide all the optimal growth condition. That means the optimum temperature, optimum pH, required amount of substrate, required amount of oxygen, and the minerals and the salts, vitamins, whatever things are essential, everything will be provided by this bioreactor. And they will produce all the product, including our product of gene of interest also. And this kind of bioreactors are the bioreactors we are using for the culturing of them, thereby to get the product. The last one, how to get the product, because the culture contain all other different type of chemical products are there, from which we want to identify the product of our gene of interest. And the process consists of it's called as normally it is a downstream process. It is called as downstream processing also. This downstream process, what are the product? Like we can say the enzymes or the alcohols or antibiotics, whatever things are there, everything will be there in that particular the product system. We want to separate them. And this kind of the separation and thereby the purification of this product commonly collectively called as downstream processing. And after that one, what is happening, the product cannot be directly sent to the market, and there should be some processing will be there, in which what we can see means and here the process, downstream processing, and that means the purification and the finishing work will be different from product to product. For example, in the case of therapeutic or the product we are using in the field of treatment, it cannot be sent directly. So that what is happening, we will be treating them with some preservative as far as the food components are concerned. And at the same time, if it is the medical component, what will be, it should go to the clinical trial. So thorough clinical trial should be there in the case of medical product. And at the same time, if it is in the food product, it should be added with the preservative, then only we can use it. Thank you.